Welcome, welcome back to the class. Now we continue with Mendelism in this class. The contribution of Mendel to genetics is called Mendelism. We know that Gregor John Mendel is called as was called as father of genetics. Gregor John Mendel contribute a lot for the new branch in the science called genetics. Modern genetics basically depends the classical work carried out by Mendel at first. Mendel was the first person who scientifically conducted experiments in plants, especially hybridization in plant at first. Nobody else before. Mendelism, it includes all concepts brought out by the Mendel through his original research on plant hybridization. Today, the modern genetics, which laid his strong foundation with the Mendelian concepts. Mendelian genetic concepts are basic to the modern genetics. Because of his contribution to understand about the inheritance of characters in plants, he was known to call as father of genetics. Gregor John Mendel is called as father of genetics. Let us see a small history about father of genetics, Gregor John Mendel, 1822 to 1884. The first genetist, Gregor John Mendel, who unraveled the mystery of heredity, who just opened the knot of mystery of heredity at first. He was born on 22nd July 1822 in Austria, in a small village called Heisendorf, Silesia. Now it's a part of Czechoslovakia in Austria. After the school education, later he studied botany, physics and mathematics at the University of Vienna. Then he entered into the monastery of St. Thomas at Brunn, now in Austria, continued his interest in plant hybridization. In 1849, Mendel got a temporary position in a school and worked as a teacher, and he performed a series of experiments. In 1856 to 1863, nearly 8 years, did his historical important in hybridization experiments on pea plants. Mendel discovered the principles of heredity by studying the inheritance of 7 pairs of contrasting characters of pea plant in his garden. Mendel crossed and catalogued nearly 24,000 and 34 plants through the many generations. After 8 years experiments, he presented a paper entitled called Experiments on Plant Hybrids in meeting of Natural Science Society of Brunn in 1866. Mendel was the first systematic researcher in the field of genetics. In he is celebrated as now father of genetics. Let us continue. Mendel was successful because there was a question. Mendel was successful because you can see Pisum sativum plant. He is doing some experimental work on the plant. Okay. Now let us see. Mendel became successful person in his experiments. The reasons for the Mendel's success. It's very important to understand. He applied mathematical and statistical methods to biology and laws of probability of his breeding experiment. So application of mathematical and statistical methods in breeding experiment was enormous. He followed scientific methods, maintained accurate data, and detailed records of his experiments. Both quantitative data, outcome of his crosses. His experiments are well carefully planned and large number of samples he used. The pairs of contrasting character which are controlled by factor were present on separate chromosome. Parents selected by Mendel were they are all pure breed lines, and the purity of the gametes was tested by self-crossing of the progeny for many generations. For many generations. That's what he became very successful in his hybridization experiments. You can see the Pisum sativum plant in which 7 chromosomes where the characters of the pea plant arranged in the chromosome is visible to us. So location of genes was 7 characters on the chromosomes of pea plant. Okay. Now let us come to that. Why he became successful in his experiment, we understood. What he done actually, Mendel experimental system. Why he chose garden pea plant? We already aware about the Mendel why he chose garden pea plant. Few just for your recall. Pisum sativum plant is an annual plant, and the flowers produced by the plants are bisexual. Keep remember, self-pollination require flowers in bisexual condition. It's an annual plant, shows very clear contrasting characters that are controlled by single gene separately. So characters are very distinct characters and also contrasting. Example, stem height, tall and dwarf, color of the seed, green and yellow, color of the flower, white and purple, some of them. Self-fertilization is very commonly occur in the plants of garden pea. Mendel, he used the flowers of garden pea plant for both self-pollination as well as the cross-pollination. He practiced both self-pollination as well as cross-pollination. The next, flowers which are larger in size, so emasculation is possible. What is called emasculation? Removal of stamens from the flower. So emasculation helps very easy to carry out cross pollination because it is very important for the hybridization. Okay, children. So emasculation is a process of removal of male sex organ from the flower. Okay, children. So now we will back to the class. What we discussed today, we discussed the definition for hereditary variation, what's meant by continuous and discontinuous variation, and also we discuss the importance of variation in this class. So in the next class we are going to continue mental experiments on pea plant. Thank you.